Right, well, it's great to see everybody. So many people here to come to honor the career of Dr. Peter Thomas, who's about to retire and leave us from the marine science faculty. But it's great to see everybody and lots of old faces from previous times. And anyway, I guess we'll have some pictures of Peter and his students in the background here. But anyway, I just wanted to make a little introduction for Peter. And so, of course, Peter has been a wonderful asset to us here at the Marine Science Institute. So he's been here, I think, since 1980. So even longer than Ken Dunton and myself. So anyway, so I just want to have some prepared things about Peter's career here. So Peter holds a primary degree in zoology and a PhD in neuroendocrinology from the University of Leicester in England. Following his postdoctoral training in fish endocrinology, endocrine toxicology at Texas A&M University, he joined the University of Texas Marine Science Institute in 1980. Peter was, I think, the first full professor in our department. He was promoted to full professor in the Department of Marine Science and Integrative Biology at the University of Texas at Austin in 1990, and he holds the HEB Endowed Chair in Marine Science. He's published over 300 peer-reviewed papers and 30 book chapters on a wide range of topics in fish reproductive endocrinology and the impacts of environmental stress on fish reproduction, and on steroid hormone receptor biology and their functions in fish and mammals. He's published numerous top-cited papers in endocrinology, including several since 2020. I don't know how much you know about Google Scholar H indexes, but his is 88, which is pretty darn good. So Dr. Thomas's primary research interest over the past 25 years has been on rapid non-genomic action of sex hormones in fish and mammalian models. His laboratory has pioneered the identification of the novel receptors of the cell surface that mediate rapid actions of sex steroid hormones, including membrane progesterone receptors, known as MPRs, that they discovered in fish ovaries in 2003. A membrane estrogen receptor identified as in human breast cell cancer cells in 2005 called GPER, and an androgen, you can tell this is not my field, right? Androgen membrane receptor they discovered in fish ovaries in 2014 that they're named ZIP9. His research group has identified the critical roles of these three steroid hormone receptors in the regulation of oocyte maturation in fish and in the regulation of fish ovary follicle cell death. A current research focus is on the structure, ligand binding, and signaling functions of membrane progesterone receptors. Research efforts in fish have been on the functions of membrane progesterone receptors in sperm motility and in human cell models on neuroprotection in Parkinson's disease, in peripheral nerve regeneration, and in mediating beneficial progesterone effects in the vascular system. So not only has his research involved fish, but also extends on to human health impacts. Another ongoing research emphasis is on the signaling and cellular functions of androgen membrane receptors in breast and prostate cancer cell lines. The discovery by the Thomas Research Group of the identities of all three sex steroid membrane receptors in vertebrates has opened up an entirely new avenue for research on rapid steroid hormone actions and functions in health and disease that have stimulated extensive research on these topics. Dr. Thomas has received several honors for his contributions in comparative endocrinology. He was the recipient of the Irving I. Geschwin Memorial Lecture in Comparative Endocrinology in 2006, and the Howard Byrne Distinguished Lecture in Comparative Endocrinology in 2008 from the Division of Comparative Endocrinology and the Society of Integrative and Comparative Biology. He gave the Gordman Byrne Memorial Lecture in Comparative Endocrinology in 2019 and was elected a Fellow of the North American Society of Comparative Endocrinology in 2023. He was also awarded an honorary doctoral degree, Doctor of Philosophy degree in biology from Örebro, I think, University in Sweden. Am I pronouncing that correctly? Anyway, he has served as chairman of the Department of Marine Science on several national and international advisory committees as an expert on the effects of environmental contaminants on reproduction and endocrine systems in fish and wildlife. Dr. Thomas currently serves on 6 editorial boards for scientific journals, including General and Comparative Endocrinology, the Journal of Experimental Zoology, Frontiers in Endocrinology, and the Journal of Steroid Biochemistry and Molecular Biology. He's also had a great impact as a mentor, and this is Sally's count. So according to has had 15 master's students, 14 PhD students, and 17 postdoctoral students that he's mentored. So he's just had a fantastic impact on the field. So Dr. Thomas, would you like to give your lecture?
Hello, this is on, you can hear me? Good, great.
Yes, please. Put the switch down.
Well, thank you, Ed, for that kind introduction. And good evening, everyone, and thank you for coming. I really appreciate everybody coming to listen to my lecture and to see some old faces from people from my lab. This is wonderful. So when I was asked to give this lecture on 43 years of reproductive endocrinology studies, actually, I thought I had 20 graduate students, and maybe I only had 19. 20 graduate students, nearly as many postdocs, and over 300 papers. I thought, well, I can't possibly summarize that in 1 hour. And we've got lots of beer and wine out there, so I didn't want to delay the process too long. So what I'm going to talk about is I'm just going to talk about a few subjects. They're going to be like postcards, vignettes, and I'm going to talk about the people that are involved with them and the major papers. And if I leave anybody out, I'm sorry. It's just I couldn't talk about all the topics we've covered. So, and the graduate students probably don't like this because they had to learn this from my class, but this is a model of the invertebrates of the system controlling reproduction. And it involves the brain, the pituitary, and the gonads. And this is what is discovered in mammals and in humans, but also there's a lot of work being done in freshwater fish. And when I came here just after Hurricane Allen in 1980, I looked at what had been done in marine fish, and none of this was known. So I thought, oh, I've got a career's worth of work here. Good. So I'm just going to go through the system briefly. So in all seasonally breeding animals, including fish, seasonal changes in photoperiod and temperature send information through sense organs to the brain, the ancient part of the brain, the hypothalamus, and then they're transduced into a peptide hormone, GnRH, through a variety of neurotransmitters. So this gonadotropin releasing hormone, which is almost the same as the one in us, in humans, this peptide hormone acts on the pituitary to cause the release of gonadotropins. And these gonadotropins are released into the blood and go into circulation to the testes and ovaries, where they cause the process of gametogenesis, the production of new oocytes, eggs, and sperm. And they do that through the production of androgens in males, estrogens in females predominantly, and progestin hormones in both. So these sex steroids then regulate many of the processes. And they do so by activating nuclear receptors, abbreviation here, the estrogen receptor, the progesterone receptor, the androgen receptor, and also binding to special globulins in the blood. So these receptors then regulate the functions of these steroids in the ovary and testes to produce gametes, eggs, and sperm. But in all egg-laying vertebrates, they also act by binding to an estrogen receptor in the liver to cause the production of yolk, which is then sequestered by the growing oocytes. So this occurs in all egg-laying vertebrates. So they also cause feedback. So the ovary and testes send information to the brain, telling the brain how developed they are through these sex steroids. So you can see it's a very complicated system. And not surprisingly, is easily disrupted by stressors, including toxic chemicals, a lot of work being done on that, and physical stressors like captivity, and perhaps also hypoxia, although nothing was known about it. So we started to study this system, and but first of all, we started to work on temperature and photoperiod changes. And then we looked at the effects of stressors like toxic chemicals and hypoxia, and how they acted on the brain to cause the production of neurotransmitters, which then regulate GnRH secretion. So the GnRH we looked at, and we also measured luteinizing hormone for the first time in marine fish, and their actions on androgen, progesterone, and estrogen production, on the production of gametes, eggs, and sperm, and the estrogen receptor in the liver, and the yolk protein production, really important part of the control of fish reproduction. And we also looked at feedback of these by these steroids through the characterization of these receptors, the first characterization of these receptors in fish, widespread characterization. So I'm going to talk about some of these. I'm going to do little snapshots talking about what we did in some of these areas. But then the second part of my talk, I'm going to really focus on what I think is the most important contribution of my lab, the discovery, structure, and functions of these membrane steroid receptors, MPRs, membrane progesterone receptors, membrane estrogen receptors, and the membrane androgen receptor. And I'm going to finish up by talking about how all these three receptors work in a single tissue in the fish ovary, and how they regulate oocyte maturation. They don't regulate sperm motility in the fish ovary, but they do in the testes. And then I'm going to be talking about membrane steroid functions in one tissue. And I'm hoping to convince you then that these steroid membrane receptors are critically important for physiological functions in important reproductive tissues. So when I first came to Port Aransas, then I worked in the mariculture lab. And this is an early picture in 1983. Here's Connie Arnold, who's a director. I worked with Joan Holt, and there's me. It was a great place to work. We're a very happy family. And I got Nancy Brown, who, if you went to the seminar recent seminar by Joe Griffith, he featured her work. She's still been working very recently. And my first graduate student, John Trent. And then I was lucky, I was able to bring NSF money with me from the IDOE program, chemical ecology, on toxicology. So I had a postdoc, Wayne Fulk Warford, who worked on that. So when we first went there then, we began our studies on reproduction in spotted sea trout and croaker. And we also did aquaculture research, induction of spawning with GnRH. And we did some work, continuing my work on stress, on stress responses of fish in captivity. And we got some really highly cited papers by Laurie Robertson, one of my first graduate students. So we did a lot of work on marine toxicology and began these biochemical endocrine responses as well. So the first thing I want to talk about is the control of reproduction, which is by the neuroendocrine axis, the brain, because it's critical to understand that if you want to understand how reproduction is controlled at all. And started these studies early, and Paul Copeland, a postdoc in my lab from the UK, went and first of all purified gonadotropin from croaker. So he went down to the dock at the Port Aransas dock where there was filleting fish and collected the heads from 4,500 croaker. It was a messy job. He had to get some of them out the bin where they would dump them. So he purified gonadotropin from croaker and developed the first radioimmunoassay for luteinizing hormone in any marine fish. And we published that in 1989. Now, reproduction in fish and many organisms is under very tight control, and it's inhibited unless the conditions are exactly right for spawning. So for example, in carp, they have to have floating vegetation in order to reproduce, to spawn. So there's a strong inhibitory mechanism by dopamine preventing that production of gonadotropin-releasing hormone. But what Paul found was croaker, which spawn out in the open ocean, don't have any inhibitory mechanism. They don't need one. As soon as they get out there, they can spawn. So this is the first example of why you need to study broadly in marine fish, because what you find out in freshwater fish doesn't necessarily transfer. So after that, Ishar Khan, who's here today, and it's wonderful to see you, Ishar, continued his work. He was a neuroendocrinologist with a lot of training. He looked at the transmitter which actually stimulates gonadotropin secretion, tryptophan, serotonin. And this is a section of the brain, a cross section, and there's a very talented neuroanatomist. And he showed in the same region of the brain, the hypophysiotropic area, the preoptic anterior hypothalamus, has both GnRH, that peptide, and serotonin. So they're closely linked. Now, we were funded by National Institute of Environmental Health Sciences for a long time, because we did some of the first work on neuroendocrine toxicity in vertebrates. And these are some of the chemicals we use. And what Ishar found was that the enzyme that makes serotonin, the stimulatory neurotransmitter, TPH, was inhibited by pre-exposure to PCBs. So PCBs inhibited this stimulatory neurotransmitter. And they also, as you see here, inhibited the production of that releasing hormone, gonadotropin releasing hormone. So it's clear then that, that the brain is an important site for disruption of the endocrine system. And also another person who's here is Sonia Matthews. And we continue doing our work on the normal system, not just the toxicology. And she showed that progestin have feedback on the brain to regulate LH secretion. So these studies then shown a lot of the toxic effects of chemicals on the reproductive system. And then the other study where we showed toxicological effects on the reproductive systems was with hypoxia. And this is this has been done in collaboration primarily with Dr. Sadia Rahman. We published a lot of papers together, 21, and it's still ongoing. And he and I went to an estuary in Florida, which was hypoxic, and with the help of James Kuma, we found for the first time that hypoxia really suppresses the reproductive axis, and it happens through the same mechanisms I showed you earlier, primarily through the brain. And so this paper was very highly cited because we did controlled laboratory studies and saw exactly the same response. So we had a pattern of changes like a clinician would have for disease that was significantly the same when we did hypoxia in the laboratory. So that was very convincing to many people, and that's why that paper's been highly cited. And then a few years later, we decided to look at a much bigger picture. So we looked at the Gulf of Mexico. And in the summer, of course, in the northern Gulf of Mexico, this hypoxic zone shown in color here spreads from the Mississippi Delta all the way over to Texas, up to 20,000 square kilometers. And we went out in the RV Pelican, and this was a lot of fun. We really enjoyed doing the field work. And we collected croaker both from the normoxic sites here and across this hypoxic zone. And what we immediately saw was the ovaries and testes at the normoxic sites, where the oxygen levels were normal, were fully grown. But at these hypoxic sites, the croaker ovaries and testes were dramatically reduced. A dramatic change. And Rahman's is a very detailed investigator, really able to look at the fine details. So he looked at all the slides of these ovaries and testes and found that in some of the ovaries, there's sperm. So there's masculinization of the females in these hypoxic sites. And we did a lot of measurements, and we showed that, in fact, the enzyme that produces estrogen was inhibited in the ovaries at these sites. So that's the likely reason why you get all stages of spermatogenesis next to the oocytes in the ovary. So Rahman's done a lot of other changes. He measured all these effects here, and he showed, including epigenetic changes and DNA methylation. And we also had these results analyzed by a number of members of the team, a population modeler, and Kenny Rose, and he showed that these reproductive effects would cause long-term declines in the population. Now, what's exciting to us is these changes in reproduction in croaker have been adopted by NOAA and other government agencies in their restoration plan for the Gulf of Mexico hypoxic zone and the ecosystem. So in order to restore the ecosystem, the reproduction in croaker has to be restored as well. So it's having an impact on management. So steroid hormone, another vignette. Steroid hormones then work by binding steroid hormone receptors. And our knowledge of steroid hormone receptors and how they work is critical to understanding how these hormones act, okay? So one of my first PhD students, John Smith, introduced us to steroid receptor measurement, and he measured the estrogen receptor in spotted sea trout. He did actually studies in the field showing changes in these receptors. One of the first studies ever to show changes at steroid receptor levels in the field, in the liver, associated with vitellogenesis. And then other PhD students, John Pinto worked on the progesterone. These are all nuclear receptors which regulates ovulation. Todd Sperry worked on two nuclear androgen receptors with different tissues, and worked with endocrine disrupting chemicals. It's pretty highly cited. And then Charles Lady worked on these proteins in the blood to bind these steroids when they circulate in the blood. But Beth Hawkins, shown here, she's from Texas, you might notice. But actually she wore this hat because she was taken by the dean then at that time to the rodeo in Houston. But actually she's from Corpus Christi and then one of the UT graduates who worked in my lab. So we then for the first time embarked into trying to these were all biochemical studies. We wanted to find out if we could clone estrogen receptor alpha in croaker. And this is our first time. So she worked on it, but she found evidence of a second receptor. And we didn't believe this because it was our first time. We kept doing it, spent over a year redoing it. And then we got trumped by Aki Gustafsson at the Karolinska Institute because he found the second estrogen receptor in rat, which we'd already found. So there's a salutary lesson there, and you have to be bold, I guess. But luckily, Beth found a third receptor, and it got highly published. So we got a lot of publications. It's published in PNAS. And what she found, this shows a distribution of ER alpha, ER beta, and ER gamma. These are all in that part of the brain where the GnRH is, which controls reproduction. These are in situ hybridizations. And you can see they're slightly different, but they're all in the similar area. So subsequent to that then, a follow-up study was, what is the relationship between these two? And what we obtained evidence was that they're quite similar. And in fact, perhaps ER gamma is due to the genome duplication that occurred in fishes, early in the evolution of fishes. And so the question is then, are these, when you get a new gene, does it do new functions, or does it just do some of the old functions of the ancestral gene? And what she did find was that ancestral ER beta had the distribution of these receptors in the nuclei in different parts of the brain. But when you looked at ER beta and ER gamma in Croker, they didn't have any new locations. So it suggests then that the original function of ER beta in the ancestor was split between these two things. And it's called subfunctionalization. They're split between these two new receptors. But there's interest, so it sounds as if nothing is new. Nothing is particularly interesting, but there was something particularly interesting because the estrogen binding characteristics of this novel receptor, ER gamma, was very different from that in the other two receptors. Different ligands bind to it. And we did some research on that, and she identified certain ligands, shown in red here, which were quite unusual and different. So we mutated those amino acids and found that, in fact, we could restore the similar binding as we see in these other two receptors by mutating that. So that was very interesting. So these are all nuclear receptors. They're called ligand-activated transcription factors. So the steroids activate these receptors to cause transcription. And this is how they work. This is a classic genomic mechanism of steroid action. The steroids enter the cell, bind to these intracellular receptors, causing their dimerization, translocation to the nucleus, where they bind to hormone response elements on gene, causing changes in DNA and RNA and transcription and translation. So this causes new protein and RNA synthesis. It takes quite a long time. So these actions are typically fairly slow. But it's been known for some time that many steroid actions are much too rapid to be explained by this mechanism. And evidence has accumulated that steroids can also act at the membrane by an unknown membrane receptor to cause intracellular responses, which are very rapid. And often they do not include regulation of genes themselves. It's a non-genomic mechanism. And the identity of these membrane receptors was unknown, but it's been known for a long time that they regulate this process. So this is a non-classical, non-genomic mechanism of steroid action. And an example of this is progesterone induction of oocyte maturation in amphibians, which has been known since the 1970s. But despite a lot of work, so anyway, what we did is we tried to see if the same thing happens in fish. So this is progesterone induction of oocyte maturation in sea trout and croaker. So first of all, does the steroid act on the cell surface? So we incubated the eggs with the steroid, with a progesterone hormone, and this is what they look like once they've undergone oocyte maturation. So they're matured. But if you inject these oocytes with the same hormone, there's no maturation, which means then that this hormone must be acting on the cell surface, not by that other steroid receptor. And this work has been done by many others, including Dr. Tokamoto's, who's here, is former professor who just talked to me by Zoom before this meeting, and Professor Nagahama, one of the pioneers in this field. So the second thing we thought was, well, if it's by this membrane receptor, it could be a non-genomic mechanism. So if you add a transcription inhibitor, this was actually actinomycin D, it didn't block oocyte maturation. So this is a non-genomic mechanism. And thirdly, we looked, if it's a membrane receptor, they all act through G proteins, nearly all of them. So we wanted to see if a G protein is involved. So this is GVVD induced by the maturation-inducing steroid. And when you add an inhibitor, pertussive toxin, which inhibits an inhibitory G protein, you block it. So it involves in a G protein, an inhibitory G protein. So it suggests then it's likely through a membrane receptor. A couple of research advances which are really important. First of all, my first graduate student, John Trent, discovered a new hormone, a new vertebrate hormone, 17-20-21-trihydroxy-4-pregnine 3O. This had not been described as a hormone in vertebrates previously, and is since shown to be the progestin that causes oocyte maturation in a number of marine species. So this is found in perciforms of marine fishes. And then a few years later, Rinaldo Patino, who's here at this seminar today, and we've worked a long time together. We published 13 papers together. He produced the first receptor assay for the progestin hormone that regulates oocyte maturation in fishes. And this is particularly important because others, until then, people have been unsuccessful in developing an effective assay. So this is really useful. And what he found was that the binding characteristics of that receptor are different from that nuclear progesterone receptor that we'd characterized previously, suggesting then it's not through a nuclear receptor. It's through an unrelated membrane receptor. But despite extensive evidence by many labs around the world for over 25 years, nobody was successful in identifying the membrane receptor. And failure to identify any of these membrane receptors really prevented people accepting this entire mechanism of non-genomic steroid actions, or non-classical, as they're called. But then in the last 20 years, 7 to 8 transmembrane receptors have been identified on the cell surface, which mediate these non-genomic steroid actions. First of all, we showed the membrane progesterone receptor. We discovered that in C-tau ovaries in 2003. And 2 years later, we showed that an orphan receptor, GPR30, was in fact a membrane estrogen receptor. We found this in breast cancer cells. And the same year, another group got similar results to us. So the two labs then had the same evidence that this orphan receptor is a membrane estrogen receptor. And then in 2014, we showed that a membrane androgen receptor, we've discovered this in croca ovaries again, was in fact a zinc transporter. So for the remainder of my talk, I'm going to talk about just these receptors. So first, the membrane progesterone receptors. And a brilliant scientist, Yong Zhu, who's here today, and hopefully you can meet him, after 6 years of intensive work and very stressful, he identified the membrane progesterone receptor on C-tau oocytes. And then in a companion paper, the human genome had just been published, we showed that in the humans and in other vertebrates, these receptors are also present. So this is the first paper showing that progesterone binds to this receptor when it's expressed in E. coli system. And then the second companion paper showed there's multiple MPRs in vertebrates, human, mouse, xenopus, pig, etc. So this had a big impact on the field. And in fact, as you can see, over 1,800 citations from this work occurred. So this is the first clear identification of a membrane steroid receptor in any vertebrate. So how do you prove this is a receptor? So that's a big problem because a lot of people did not believe this. So the first thing you have, so these are the criteria. First of all, it has to have a structure. These are transmembrane domains. This structure is typical of a membrane steroid receptor, of any membrane receptor. Secondly, remember that we showed that progestins activated oocyte maturation on fish oocyte. Do you remember that? So clearly then, we found it on the oocyte, which is known to mediate these non-genomic actions. And thirdly, in E. coli, it binds specifically progestins, and this shows a couple of progestins binding to this receptor expressed in E. coli. The next criterion is it has to signal. Remember, these are membrane receptors that have to signal inside the cell. So what we've showed is this is proximity ligation analysis. And these red dots show that the MPR is within 40 nanometers of a inhibitory G protein. So it activates and is coupled to an inhibitory G protein. And consistent with that, inhibitory G proteins decrease cyclic AMP levels. And here you can see that the maturation-inducing steroid decreases cyclic AMP levels, which is blocked when you inhibit that G protein. Other criteria, well, of course, it causes this hormonal regulation of this receptor regulates oocyte maturation. So the MPR is upregulated during oocyte maturation, completely consistent with its function in regulating this process. And finally, oocyte maturation by this steroid is inhibited when you knock down expression of MPR alpha. So on the basis of this, then, there's a lot of people that moved forward and did additional studies on MPR alpha. But there was a lot of people antagonizing our early results. So we did a couple of additional papers, highly cited papers. We talked about the membrane receptor, and we kind of critiqued some other people's results, which were negative. And then we did another paper in endocrinology showing that when you express these receptors in a more physiological system, in other words, in a human or vertebrate cell line, this shows that in sea trout, that maturation-inducing steroid has a highest affinity for the receptor. Whereas in human, the progesterone, which is a human progestin, has the highest affinity for the receptor. So the specificity of these receptors is completely in agreement with their biological functions. And then we showed more clearly that MPR activates these inhibitory G proteins, which is observed, which is consistent with us showing that cyclic AMP was downregulated. So in our original paper, Young and I also identified MPRs on sperm, on crocus sperm. And this shows the motility of crocus sperm before any hormone is added. So 1 minute after you had 20-BDS, that maturation-inducing steroid, you can see sperm motility is greatly activated. Now, this is really interesting. This is the first evidence that progestin stimulates sperm motility. And this, of course, is what we see in mammals. In humans, progesterone stimulates sperm motility. But this is the first time it's been shown in a fish. So this early work was done by Chris Tubbs, and also later with Wenxian Tan, who's here today, and currently with Caroline Matkin and Teresa Bennett, who's still working on this. And I will also mention Yossi Azen, who couldn't be here today, but I'm still working with him on this project. So we found MPR on crocus sperm, and here it is on flounder sperm. This is the head of the sperm. This is the midpiece. It's on the flagella. And interestingly, it's also on human sperm, but it really hasn't been fully explored. So it's a very complex system that Chris and Wenxian described. It involves a stimulatory G protein now, which causes an increase in cyclic AMP. That's one mechanism through the alpha subunit of this G protein. And then the beta gamma subunit activates epidermal growth factor, phosphorylation of ERK, and also it involves a PI3 kinase phosphodiesterase pathway. We're still studying this pathway. Teresa's studying this in the flounder now. And Caroline, who's just finishing up these experiments, showed that it increases calcium levels in the sperm, and these increases in calcium in the sperm directly stimulate sperm motility and fertility. So this knowledge of this activity of flounder sperm by progestin provides a new methods to promote male fertility in finfish aquaculture. But most of the work I've done over the last 15, 20 years has been on mammalian cells. I've probably published about 50 papers on that. Now, an interesting thing is during pregnancy, progesterone levels are high in women so that the uterus does not contract and there's an abortion. It's well known. And it acts through the nuclear progesterone receptor. But when at term, of course, the uterus has to contract to cause birth. The problem is progesterone levels are still high. Nobody could understand why. Well, working with Emmanuel Kataris at Brunel University in England, we got human uterine muscle cells, and we showed that at the end of the reproductive system, during labor, the amount of MPRs on the surface of these cells increases, and progesterone acting through these MPRs shuts off the nuclear receptor by decreasing the amount of the co-regulator that regulates this receptor, SRC2. And the other thing it does was it increases the production of a contractile protein, myosin light chain reaction. So this has had a big impact, and a lot of people have followed up on this because it's a big question in human reproduction. We also, with Linda Goudis from Stanford University, was looking at the immune function through MPRs in T lymphocytes during pregnancy, because these T lymphocytes did not have the nuclear receptor, but we know that progesterone regulates it. Luca Castelnova, who's here today, he came here as a PhD student from Milan University and came back as a postdoc. And he's been looking at peripheral nerve regeneration, repair, and also in Parkinson's disease. And he showed that, that it has a protective effect and a regenerative effect through MPRs. And then Gwen Dressing, who was a PhD student in my lab, and looked at how in breast cancer tissue biopsies, the levels of some of these membrane progesterone receptors are upregulated. And two other visiting PhD students, Maria Cavalletti from Argentina and Basar Sinrai from, where was it? Forget where exactly it was. Oh, anyway, from Serbia, I think, from Serbia. They looked at the effects of MPRs in other cancers, prolactin cancer and also endometrial cancer. So if you think about membrane progesterone receptors and rapid steroid actions, you would expect the brain to be a target. Many things happen very quickly in the brain. And Dr. Yeffy Pang, who I've published over 50 papers with, looked at this binding, the distribution of steroid membrane receptors in the human brain, and also the functions of them. And what he found was that this form of the membrane progesterone receptor, which I haven't talked about yet, MPR delta, is highly expressed in lots of parts of the brain. And interestingly, this receptor binds a neurosteroid, which has really important functions of the brain, allopregnanolone. You can see the binding curve there. And allopregnanolone, through this receptor, decreases cell death. And interestingly, a drug that's been under development for epilepsy, galaxolone, also binds MPR delta. So here we have protective effects through MPR to decrease cell death, which is really important, this cell death, because it occurs with aging, Alzheimer's disease, multiple sclerosis, and many other diseases, brains and the neuroendocrine system. And then galaxolone which is an anti-epileptic drug. And I think it's already finished stage 3 trials, clinical trials. Not only does it prevents epilepsy, but it also prevents cell death, which is really important because that's another problem with all of these neurological diseases. So most of these studies have been done with MPR alpha. And I'm just listing the studies that have been done in my lab and with collaborators. You can see there's many functions. And importantly, though, there's many cancers, there's many deleterious functions. So despite all this work by us and an equal number of studies by others, we still don't know the structure of the steroid binding domain of MPR alpha. And that's the final criterion. All the nuclear receptors, they know the structure of the steroid binding domain. So this was not known. And this is really necessary for its final criterion as a steroid membrane receptor, and also to develop specific drugs that only target these MPRs. So in that 2007 paper, we showed that these 5 members of MPRs belong to the progesterone and adiponectin receptor family. This is the family. These are the MPRs here. There's 11 members of this family, and they're related to hemolysin 3 receptors. They're urobacterial origin. They're not related to GPCRs, those G protein coupled receptors. But the 3D structures of MPRs is unknown. We didn't know the structure of any of these. And this is a problem because membrane receptors, it's really difficult to find their 3D structure. In fact, if you look at the protein data bank, only 1% of the proteins in the protein data bank are membrane receptors. So homology modeling of protein structure based on the known structures of closely related proteins is an effective alternative when the 3D structures are known. And the X-ray structures of adiponectin receptors has recently been found. And interestingly, so they're in the same family, and interestingly, there's many conserved amino acid residues in these and in MPRs. So it means then construction of a homology model of MPR based on the structure of adiponectin is a valid approach. So the human homology model was developed by Jan Kelder. I've had a collaboration with him. He's a medicinal chemist in Organon, a drug company, worked with him for 20 years. And one of the big questions was what is the orientation of these receptors in the membrane? And there was a lot of controversy over this. So we adopted a different approach, the positive inside rule. And this says that positively charged amino acid residues are predominantly inside the cell. And this shows MPR alpha, and this shows a number of positively charged residues inside the cell. And with this, it shows that the N-terminal is intracellular. And this looks the same then as the adiponectin receptor. So this just shows an electrostatic map, potential map, showing the negative charges are extracellular, the positive ones are intracellular. And based on the X-ray structure of adiponectin 2, we made this homology model of MPR alpha with the N-terminus inside. So this shows the structure of the ligand binding homology model of the ligand binding domain of MPR alpha. This is progesterone in there, in the ligand binding pocket. Now, what makes the defining structure of MPRs that distinguishes them from progesterone distinguishes them from other steroids is these functional groups on the steroid molecule. So this 20-carbonyl is really important, and this 3-carbonyl. So the nuclear progesterone receptor recognizes these two, and it doesn't recognize any other steroid with other functional groups. So what we noticed was that in our model is an H-bond interaction between glutamine 206 and this 20-carbonyl of progesterone, shown in magenta. So it suggests that this is critical for binding. So how do we test this? So what we did is we did receptor binding assays with ligands that had this 20-carbonyl, and then compared it to steroids that didn't have that 20-carbonyl, the 20-deoxy steroids. And we did that, and we also looked at the 3-carbonyl, and what we found was, surprisingly, there weren't any amino acids close to that 3-carbonyl. So it suggests that unlike the nuclear progesterone receptor, this probably is not important for binding. So what we did was we tested the ligand specificity of this 3-carbonyl with that group and with steroids without that group on it to see if they found similarity. Now, as shown here in these white boxes are other steroids in the ligand binding domain, which are supposed to have, in our model, had hydrophobic interactions with progesterone. These are aromatic and aliphatic amino acids. So what we did in addition to these ligand binding studies, we also mutated these key residues in the binding pocket, valine 146 and also that glutamine 206. So first of all, I'll show you the ligand binding specificity. So this is a competition curve. So what you have is you have radioactive progesterone binding to the receptor, and as you add more of the cold ligand, progesterone, you displace increasing amounts of the radioligand. So this shows then, with this cold progesterone, it's a very strong competitive binding. But when you remove this 20-carbonyl in this compound here, P420, there's no displacement. So this compound does not bind at all to the receptor. Looking at the 3-carbonyl, so it's the same thing then. Progesterone binds very well. But when you remove this 3-carbonyl here in this compound, it also binds really well. So the 3-carbonyl then is not critical for binding to the membrane progesterone receptor. And this is the same results with androgens. So the ligand specificity then is consistent with this homology model prediction. And the important thing is this provides a basis for designing drugs that activate NPR, which lack this 3-carbonyl. They'll bind to NPRs, but not to the nuclear receptor. And the mutagenesis results showed similar things. So this shows binding with the wild-type receptor. When you mutate valine to aniline, you decrease the hydrophobicity, and therefore you decrease binding and also serine. So that hydrophobicity of the wild-type valine is really important for binding. When you reproduce it with another hydrophobic, leucine, you restore binding. So it shows then that hydrophobic interaction in the ligand binding product is critical. And similarly, for glutamine 206, it's a polar, it contains H bonds. When you mutate it to non-polar compounds, non-polar amino acids, you lose binding, but with asparagine, and it's polar and H bond donor, you can partly restore binding. So it provides further support then for this homology model and the critical criterion for receptor designation. And homology modeling, then the construction is incomplete, but it really narrows the gap between sequences and structures. And we're hoping with more new methods like cryo-electron microscopy, we can solve more protein structures of these membrane receptors. So I'm going to finish off by just talking about essential functions of the steroid membrane receptors and just take one example, ovarian functions in fish. And I'll start by talking about oocyte maturation. And this work is possible. We could do this all year round because Susan Lawson, who's here tonight, and her husband, Woody Lawson, looked after our fish and were able to cycle them so we could take oocytes throughout the year to do these experiments in oocyte maturation. And other people are involved with Sham Prakash and Caleb Harris. And Dr. Toshinobu Tokumoto is here today, came all the way from Japan, and I'm really delighted he's here. And he continued these studies. He did some work in my lab and continued these studies with a goldfish model and a zebrafish model afterwards, and has had some very exciting results. So this shows what the eggs will look like. This is a fully grown oocyte surrounded by follicle cells. This is undergoing oocyte maturation, these different stages. And then this is a final stage. And at this stage, they can be fertilized. And Ronaldo Patino showed that there's a two-stage model of oocyte maturation. The first stage, the follicle cells became able to produce 20-bit-S. The enzymes that make 20-bit-S are induced, and it's a genomic mechanism. Then the second stage, where 20-bit-S is reduced and oocyte maturation is induced, which I've already shown is a non-genomic mechanism, is the second part. And this paper has been highly cited. And then another graduate student in my lab, Margaret Pace, who's had a lot of experience in signaling, showed more definitively that NPR is coupled to an inhibitory protein, causing cyclic AMP decrease. And this is a likely mechanism inducing oocyte maturation. And she also showed that it activates the PI3 kinase AKT pathway to induce oocyte maturation. And these are all the species we studied oocyte maturation in. But we had a surprising discovery. We found that estrogens prevent oocyte maturation. And there's been a lot of work done on oocyte maturation by many labs, but really had never been documented. And here you can see that estrogens decrease oocyte maturation, but when you inhibit the production of the enzyme that makes estrogens, you get a lot of spontaneous oocyte maturation. And then if you remove those follicle cells around the egg, you can decrease the production of cyclic AMP. So cyclic AMP seems to be maintaining oocyte meiotic arrest, as they call it. And when you remove those follicle cells associated with this decrease in cyclic AMP, you get an increase in oocyte maturation. So this is really interesting. This is the opposite effect for the progesterone. It causes increases in cyclic AMP. And we wondered whether this is mediated through G protein, GPER, or GP, or as it was known, GPR30. So we discovered GPR30 or GPER, as it became known later, in breast cancer cells. It was an orphan receptor in 2005. And as I said, shortly after, Eric Prosnick's group got the same results. This is my most highly cited paper. And these studies by us and him, we show that it activates a stimulatory G protein. It's a classic GPCR with 7 transmembrane domains. And we found that these nuclear estrogen receptor antagonists are in fact agonists through the GPR30 or G. And Eric Prosnick found a specific agonist G1. So this just shows the binding. This is saturation curve, and the scat has very high affinity for the receptor. But there are, of course, a lot of people who worked with nuclear receptor wondered about this. So I worked with Edward Falado for a long time from Brown University, and he showed that, in fact, this receptor is clearly on the cell membrane, and it's only the cell membrane that binds estrogen. So it's a membrane estrogen receptor. And since then, many people have jumped on this and worked on this. There's now over 1,000 papers on GPR in mammalian models. One thing that I wondered, though, estrogens receptors, a nuclear estrogen receptor is many xenobiotic foreign chemicals bind to it. They're called xenoestrogens. It's well known in the literature. And it's one of the major endocrine disruptions described. Bisphenol A in baby bottles, if you all remember that, is because it's afraid about effects of estrogen on young males. So we wondered whether the membrane estrogen receptors or the membrane steroid receptors were also disrupted by xenoestrogens. So a year after we published that paper, Jingdong and I, and we published 29 papers together altogether, we looked at the binding of and activation of GPR30 by these xenoestrogens. And we found, in fact, they were very effective at inducing responses mimicking the effects of estrogen. And so this paper was really highly cited, and it led to a lot of people now working on this, particularly in breast cancer models. But we also showed that other membrane receptors are involved. So this is a membrane progesterone receptor alpha. We showed Kelly Doughty. We showed that all these compounds interfere with that zinc, progesterone induction of sperm motility, and also oocyte maturation. And we also more recently showed that the membrane androgen receptor, which I haven't described yet, that the fungicides, which are anti-androgens through the nuclear receptor, also act to inhibit apoptosis through this membrane receptor. And finally, Catherine Ondrzejczyk, who's here, she studied the effects of hypoxia and found that all these receptors, NPR, this androgen receptor, which I'll describe in a minute, GPR, their signaling is knocked down in Croker during the hypoxia. And another graduate student, Amanda Fitzgerald, looked at oocyte maturation. Remember, estrogens inhibit oocyte maturation. And she looked at bisphenol A and alkyl phenols, and they mimic the effects of estrogen to inhibit oocyte maturation in fish. So these are the first evidence then for endocrine disruption through non-genomic actions through these membrane receptors. And I think it has important consequences for human health. And a lot of people now are following up on this, particularly in mammalian models. So this is just to prove that, in fact, this effect of estrogen and oocyte maturation is mediated through GPR. So here's GPR on zebrafish oocyte membranes. This is estrogens inhibit spontaneous maturation in this other model. They also decrease progestin-induced maturation, shown here. And this effect is mimicked by this GPR-specific agonist, G1. So this shows oocyte maturation in response to progestins. These clear eggs here are the ones that have undergone oocyte maturation. But when you add G1, you can see it's dramatically reduced. So, so then we did show that DHP, or these progestins, decrease cyclic A and P production, whereas these estrogens increase it. So that's the mechanism of inhibiting oocyte maturation. And when we silence the GPR, we silence RNA, so without the silence, without antisense, estrogen inhibits the effects of this progestin to decrease oocyte maturation. When you silence it, you block that effect. You block that inhibitory effect. And finally, Candice Payton, who's here tonight as well, did a very, looked at the, so this, all this is, I mean, talking about is through the alpha subunit of the heterodimeric G protein, but the beta gamma subunits also involved in oocyte maturation. And she showed through a very complicated pathway, including epidermal growth factor, that this also inhibits oocyte maturation. So, so it shows then that the meiotic arrest, or the prevention of oocyte maturation, is mediated through cyclic AMP synthesis and this epidermal growth factor ERK pathway. But what's interesting is there's reciprocal regulation of this membrane estrogen receptor and NPR alpha by estrogens and progestins. So if you, and we looked at, this is a protein levels, but also we looked at messenger RNA levels. So estrogen then increases the amount of the G protein. It regulates its own receptor, G protein estrogen receptor, mimicked by G1. Whereas estrogens decrease the amount of NPR. And conversely, progestins decrease the amount of GPR, but increase the amounts of NPR alpha. So if you think about it, then you get upregulation of GPR and downregulation of NPR by estrogen and its specific agonist, G1. So that helps main meiotic arrest. And then afterwards, downregulation of GPR and upregulation of NPR by progestins promotes oocyte maturation. And the fact that these specific hormones, so this is a specific hormone for NPR alpha, and this is a specific hormone for GPR, mimic the effects of the natural hormones, suggests it's mediated through NPR alpha and GPR30. So on the basis of this, we propose the following dual model of oocyte maturation. During the first phase, the vitellogenic growth and the beginning of the LH surge, you have a high production of estradiol, progestins are low, and estrogen acts via the G protein couple receptor to prevent spontaneous maturation. It involves production of cyclic AMP, and it also acts by the epidermal growth factor to increase cyclic ERK and cyclic AMP and GPMP to inhibit phosphodiesterase activity. And then we have another pathway, the natriuretic pathway, that's regulated by estrogen through GPR, which I didn't talk about. And it also acts through cyclic GMP to decrease the activity of the enzyme that removes cyclic AMP so that their cyclic AMP levels build up. And finally, we showed that through GPER, estrogen inhibits the membrane progesterone receptor. So the oocytes remain in our called meiotic arrest. And then in the second phase, oocyte maturation, the production of estradiol declines. And then actually we showed that a metabolite of estrogen, 2-hydroxyestrogen, blocks GPR action as an antagonist. And then the progestin surge is increased. This is due to a switch in the steroidogenic pathway. It stimulates oocyte maturation through inhibition of adenylate cyclase and cyclic AMP. And therefore, this downregulation allows meiotic maturation to proceed. And also the PI3 kinase AKT pathways increases the amount of that enzyme that degrades cyclic AMP, phosphodiesterase, allowing oocyte maturation to proceed. So we've looked at oocyte maturation, but also we find evidence that MPRs are also in the follicle cells surrounding the oocyte. And Abby Benninghoff, PhD student, developed an assay to measure the activity of these cells. And this is a granulosa cell. You can see on the plasma membrane, you can see labeling of MPR alpha. And what MPR alpha does through 20-beta-S, it decreases apoptosis, which is called cell death. So it helps maintain that follicle. Okay, so it decreases the cell death. And when you block it with this effect with silence RNA, MPR alpha, you can lose this effect. So it's regulated through MPR alpha. Now, Shenong Zhang, another student in my lab, looked at, oh, by the way, Gwen Dressing, who did this, showed that it also acts through ERK signaling as well as cyclic AMP. And this is the same pathway that mediates anti-cell death or anti-morbidity in breast cancer cells. So it's the same pathway in fish granulosa cells and breast cancer cells. But Shenong Zhang showed that androgens also regulate apoptosis of fish granulosa cells through a membrane androgen receptor. Which brings us to the point of looking at the third receptor, the membrane androgen receptor. And initially, Alyssa Braun or Alyssa Dott characterized a membrane androgen receptor biochemically in Croker. So we knew it was present. And then Hawkenberg led the postdoc in my lab, and others worked on discovering the identity of the membrane androgen receptor in Croker ovaries. And it was first discovered in 2014. And is a member of a zinc transporter family, the ZIP family. There's 14 members of this family. And what they do is they transport zinc into the cytoplasm from the extracellular compartment or from intracellular compartments. And this is really important because zinc homeostasis is critical in the cell. There's an equal number of receptors called ZINTs that transport zinc out. So there's altogether 25 receptors, proteins for zinc that transport zinc in and out of the cell. So we found it's a high affinity. This is a scattered and saturation analysis, high affinity receptor. And The ZIP9, this particular member of this family, is very different from all the others ZIPs. It's much smaller, and it's a sole member of a small family, subfamily 1. And it's the only known liganded zinc transporter. So it activates the G protein, and importantly, zinc transport and androgen signaling are closely intertwined. So when you add testosterone, you increase zinc in the cell. And it also, and what you find is that testosterone increases apoptosis, shown here. But the nuclear receptor agonists don't do anything. So this is the first evidence that a single protein can have both steroid signaling and zinc transporter functions. And Aubrey Converse, who's also here today, studied this in Croker and looked at early stage and late stage Croker. In an early stage Croker, she found that testosterone increases cell death, called apoptosis. When you knock down ZIP9, you lose that inhibition, increase. And we found in this early stage that ZIP9 is coupled through that proximity ligation analysis I described earlier, is coupled to an inhibitory G protein. But then in late stage follicles, it does the opposite. Testosterone increases apoptosis, and this is blocked by silenced RNA, and is coupled to a stimulatory G protein. So in the same cell, at different stages of the ovarian cycle, this same receptor can bind to different G proteins. Quite fascinating. And these effects are blocked with ZIP9s or the messenger membrane androgen receptor silenced RNA, but not with nuclear androgen receptor silenced RNA. So it suggests that these mechanisms of testosterone through ZIP9 are stage dependent. She also showed opposite effects at early and late stage of testosterone on zinc and on cyclic AMP. So this is her proposed model of cell death of granulosa cells. In the early stage, ZIP9 is coupled to an inhibitory G protein, decreases cyclic AMP, decreases apoptosis, and also these pro-apoptotic members, BAX and P53. Whereas in late stage and post-ovulation, ZIP9 is coupled to a GS, increases cyclic AMP, increases apoptosis through these and increases the pro-apoptotic members, BAX and P53. So it's all chopped off my title, it doesn't matter. So basically then, these membrane receptors have multiple functions in fish ovaries. Oocyte maturation, I've shown you, is regulated by 2 of these membrane receptors. It's inhibited through the membrane estrogen receptor, GPER, and it's stimulated through MPR alpha. So it's a dual control of oocyte maturation. And similarly, apoptosis or cell death is regulated by 2 receptors, MPR alpha, which inhibits it, and then depending on the stage, ZIP9 can either inhibit cell death or promote it. So it's very showing how important then all these processes through membrane receptors are for regulating ovarian function. And we've shown with these knockdown studies in zebrafish that the GPR is also important in fertility in zebrafish, and it also, ZIP9 has an important role in the zinc spark for egg activation, completing meiosis, and larval survival. So in conclusion then, sex steroid membrane receptors mediate numerous essential functions in fish ovaries. And I hope you can agree that these are really critical functions for ovarian, for the ovarian function. They're dependent on these membrane receptors. And also identification of these steroid membrane receptors has opened up a new avenue of steroid hormone research. Now we can look at investigations of non-classical, non-genomic rapid actions of steroids. But there's many unresolved issues, of course. I'm just going to acknowledge all the people that have worked on this. And I think Joseph Azen's listening, and I apologize, Yossi, I did not talk about PGMRC1, another story that we didn't have time to talk about here. But people shown in blue then are the major people working on this. But there's many people worked on these topics. I had many collaborators and funding from numerous sources. And all that work was due to the brilliant people I've had opportunity to work with. And I'm very grateful. So thank you, everyone. This has been a great 43 years. It's been a wonderful place to work. It really is. It's like a family. And it's nice to work somewhere where you know everybody. It really has been a fantastic place. I particularly thank Sally Palmer and Ed Buskey for organizing this event, and all the MSI staff and Jim, for example, for helping it work. And finally, I'd like to my wife for all her support during these years. But I've just got one more slide of this. And these are the MSI directors that helped me all that time. So I've outlived some MSI directors. So I'm going to finish with a video clip of the zinc spark mediated through ZIP9 in the zebrafish oocyte. And for this, Aubrey got second place in endocrine images in art competition this year by the Endocrine Society. So I'm just going to leave that. And there's that zinc spark. Thank you very much.